Welcome to the Step 1 Microbiology USMLE reviews from Kaplan. I am Kim Muscatello and I have my PhD in microbiology and immunology. I'm currently the director of the microbiology and immunology department at the Lake Erie College of Osteopathic Medicine. What we're going to do over the next several hours is review microbiology that's pertinent to the USMLE Step 1. I'm going to definitely point you in the direction of high-yield topics and information. We may be glossing over some of the lower yield facts and information. We'll throw in some vignettes where they're appropriate, and I also intend to have quite a bit of fun here. So stick with me. If I'm laughing here, I'm hoping you're laughing at home. All right. So the first thing we're going to do is we're going to look at Chapter 1, which is the General Microbiology chapter, and just really focus on some of the introductory information that is testable for the USMLE. Now, the first thing that you see is basically comparing the different types of pathogens that you're going to encounter in practice and that are going to be testable on the USMLE Step 1. And we're going to be covering all of these different groups. So again, this isn't what I would call super high yield information, but I'll just go over some of the salient points for you, and then we'll move on from there. So clearly the viruses, and then moving forward to the parasites, viruses are obligate intracellular pathogens. So technically, they are using the host cell to survive. They're basically capsizing all of the host cell machinery to replicate. Therefore, they're extremely small. You really can't see a virus on a light microscope as you see in some movies that you've probably seen. Moving a little bit further on to bacteria, you'll notice they're just a little bit larger than viruses, and these are the prokaryotes. So when you're comparing the prokaryotes to the eukaryotic organisms, there are going to be quite a few differences. And the differences are really beneficial when we start talking about treatments for these organisms. Prokaryotes, being very different from eukaryotes, are much easier to treat than the eukaryotic pathogens. Their way of replication is different. They have different ribosomal subunits that are often the target of the antimicrobial drugs. And if you just follow down on the list for the bacterial agents, you can see that the messenger RNA, in addition to being monocistronic, is polycistronic. So you can have basically several different proteins encoded on one strand of messenger RNA. Their replication is very basic. It's by binary fission. You can't get much easier than this. 1 makes 2, 2 make 4, and so on and so forth. So when we get to the chapter on bacterial genetics, we're going to show you some of the ways that bacteria can acquire different mechanisms of resistance. I'm going to shoot forward to the next slide here just to show you kind of the rest of the features for the bacteria. Keeping in mind, again, the ribosomes are somewhat smaller than you're going to find in eukaryotic cells. Again, very good target for the antimicrobial drugs. Cell membrane for bacteria basically are not going to have sterols in the membrane, but similar. It's a phospholipid bilayer, so similar to a cell membrane from a eukaryotic cell. And then, of course, the cell wall, which is found in both gram-positive and gram-negative organisms, that's made of peptidoglycan. So now if we go back and just really look at the fungi and parasites, now we're into the eukaryotic system, and you can see that the organisms are going to be a little bit larger. As far as replication of DNA, they're going to be very similar to the mammalian cell system, so they're much more difficult to treat. As far as replication goes, they're monocistronic. They're going to follow the mitosis and meiosis, as do the human cells. And again, if you continue on to the next slide looking at the fungi and parasites, you can just note that the ribosomes are a little bit larger. However, when you get to the cell membrane, what's really important to note is that the fungi use ergosterol instead of cholesterol in the cell membrane. Now that is really important because that's one of the major targets for the antifungal drugs. So that allows us to treat the fungi. That's one thing that we can find that's actually different from the fungi and the eukaryotic cells. Parasites become a little bit more difficult. So with parasites, again, they have cholesterol in their cell membrane just like our cells do. So again, a little bit more difficult to treat. So you basically just want to have an overview of the differences. While I don't think you'll get a specific question on necessarily the information in the table, knowing the differences between the ribosomal subunits, etc., and the mechanisms of replication can definitely be used in a treatment type question where they're asking the mechanism of action of a drug. Okay, so we're going to move on and talk about just a little bit about normal flora. Normal flora, as you know, is a good thing overall. We wouldn't really be able to live very well, at least, without our normal flora. They do a lot of things for us. Normal flora occupy a lot of space, not only on the skin and outside, but along the mucosa. When you have a nice, good normal flora, everything seems to be kind of maintained and intact. They know just how much space they have. They know just how much food you're going to give them. And so when a pathogen tries to get in, they really have to work extra hard to combat the normal flora. You'll know as we start going through the different bacterial agents that some of the normal flora are definitely associated with disease. And oftentimes we'll call that an endogenous infection. So we're actually acquiring an infection from our own flora. Now, when does that happen? That happens when these organisms get lost, okay? You've probably all been lost at one time of your life, maybe not anymore with the GPS, but E. coli doesn't have a GPS. So when it gets lost from the gut and it happens to get to the urethra and up through the urinary tract, it can cause a urinary tract infection. And we're going to see that kind of over and over again with a lot of these organisms that we call normal flora. They definitely contribute to the overall health, okay? So by having a somewhat acidic pH on the skin, that's going to retard the growth of some of the other organisms. They help us synthesize vitamins. And again, there's that general competition for space. They're not going to be very happy when a pathogen tries to come in and steal some of that space and steal some of that food. Now, what this table does is really just kind of shows you some of the different body sites and what typical normal flora you'll see at some of those sites. So I just want to go through those a little bit with you, and then we'll talk about some examples of when that's going to be important. The blood, of course, your internal organs should be sterile. If you find bacteria there, there's definitely signs of an infection. So that's probably one of the most important things that you can know. The cutaneous surfaces, so your skin, probably most common is going to be staph epidermidis. That's very common on the skin, but again, that definitely helps retard some of the growth of pathogenic organisms. Less notable, but can also be on the skin, and we might call them transient flora of the skin. You can find Staph aureus. You can find some of the other species of streptococci, and also yeast can be transient flora of the skin. When are you going to get infections from the normal flora with the skin is when you get a wound, when you get a cut. You can easily introduce, I got a nice little paper cut here on my finger. Those always hurt like way worse than you think they're going to hurt. So probably what I did was I introduced some of the normal flora from my skin into my hand there. And if I didn't clean that out pretty good right away, eventually I could get an infection from that. So that's an example of a cutaneous normal flora causing an infection. Now the nares are very common for Staph aureus, so you know that. So if you go to a restaurant and you see the chef picking his nose, digging in there, walk out, because you could clearly get staphylococcal food poisoning, which is probably the most common cause of food poisoning. So Staph aureus is very, very common in the nose. I'm going to show you a little bit later, but I'll show them to you now. This is our Staph aureus superbug. This is a MRSA though, so he's got a cape because he's a super hero basically for the organisms. We'll talk about him a little bit more later, but you can definitely see that it's in a grape-like cluster, and I'll compare that to strep when we get there, but he's going to make an appearance a little bit later on. Now, the oropharynx is loaded with different types of normal flora. The Viridan strep you've all probably heard of as far as causing cavities. They can also cause endocarditis if they get misplaced. They can typically find their way to a damaged heart valve. So they're probably one of the more common organisms in the oropharynx, along with, you've got a lot of other species of streptococci. Haemophilus is also very common there, but probably the non-B serotypes as far as being normal flora. You also have non-pathogenic Neisseria as part of your normal flora in the oropharynx. So when we start looking at diagnosis, we're going to talk about some ways that when we're trying to diagnose and culture Neisseria, to make actually a definitive diagnosis, we're going to have to try some way to inhibit the growth of that Neisseria that's part of the normal flora. The gingival crevices, you all brush your teeth hopefully, but it's really hard to get down in those crevices. That's why you're supposed to floss. The gingival crevices are famous for having anaerobes because there's not very much oxygen down in those crevices. And here's classic for some of the anaerobes like Prevotella and the Fusobacterium and even many different species of Streptococcus. Now, moving on to the stomach. The stomach really has no normal flora. Now, why do you think that is? All right, so I'll have to tell you because clearly you're not going to answer me. Maybe you are. I don't know. But the stomach doesn't have any normal flora, and it's because of part of your innate immunity. The innate immunity in the stomach consists of the acidity there. There's not really a whole lot that can live in that kind of acidity. So as far as normal flora, we have nothing that is just home to the stomach. Now, there is one organism that we know that can live and replicate in the stomach. So I'm going to give you a second to think about that, and I'll tell you what it is. Of course, it's Helicobacter pylori. So that's the only organism that we know that can actually live in and replicate in the stomach. And it's basically because of a virulence factor that it has called a urease that helps to neutralize the acidity. Everything else that all your agents of gastroenteritis basically are just doing one of these, like, please let me through, please let me through, please let me through. As they pass through the stomach, a lot of them are going to die on the way through, but a few are going to make it to the other side, and then they'll be able to establish disease. Now, the colon, as you might imagine, tons of organisms in the colon. And here's where E. coli got famous. As far as anaerobic species, one of the most clinically relevant anaerobes that is in the colon is Bacteroides. Bacteroides fragilis is most often associated with disease, so you just want to kind of keep that in mind. In breastfed infants, you'll also have Bifidobacterium. And you can see then how the flora becomes transient, okay? So just kind of keep that in mind. So at different times during life, your flora is going to change. The best example of that is in the vaginal mucosa, where you have a lot of lactobacilli, other organisms, including some of the streptococci. What happens when you disrupt that flora with antibiotics? Typically, you will get overgrowth of other organisms, overgrowth with yeast. You may get overgrowth with some of the normal flora there and get something called bacterial vaginosis. So the flora for the vaginal mucosa is very important, and again, it does change at different points of life, and therefore is actually transient. Okay, now, what kind of questions are you going to get about normal flora? Okay, if we go back and look at the cutaneous surfaces in Staph epidermidis, let's say you have a case where you have someone that has endocarditis. And you do 3 blood cultures, okay? And typically you're supposed to do those blood cultures during the febrile periods. You're going to do 3 consecutive blood cultures, and one of them is positive for Staph epi, okay? you're going to have to make the decision then, is it Staph epi that's causing the endocarditis, or is it maybe something else that we weren't able to look for or we didn't know to look for the first time around? If Staph epi is in one of 3 blood cultures, you're probably going to go with that it's a contaminant, okay, and it's not the actual cause of the endocarditis. However, if it's in 2 or 3 of the cultures, then you might consider it as the organism that's actually causing that infection. So that's why it's a really good idea to have a little bit of knowledge as to which organisms are going to appear at which sites, so you know that when these types of cases come up on the USMLE, that you're going to be able to say, you know what, I don't think that that's Staph epi. It was only in one of those blood cultures. It's probably got to be something else maybe we didn't look for. So keep that in mind as you're going through some of those questions. We'll come back to a lot of these later as we talk about some endogenous infections.